Welcome. My name is David Milne, and I'm the chair of Greater Manchester Humanists. And each year we hold a special meeting to commemorate the life and work of one of our most important scientists, Charles Darwin. And I'm delighted to say that this year we have as our speaker a leader in the field of neurophilosophy, namely Professor Patricia S. Churchland. She is a professor. P's are always all good words for me, folks, so you just have to hang on a minute. She is professor of philosophy. Gosh, two P's in a row, I'm hoping. At the University of California, San Diego, and the recipient of a MacArthur Fellowship. She's written a number of books, including neurophilosophy towards a
So, as we've lost David for the moment, as I'm sort of co-host, can we start off with your talk? Phil, then anything more you would have wanted to introduce yourself about?
Okay.
Well, okay?
Yeah, sure. This is just fine. I'm really delighted to be here and to be able to give you this perspective on what neuroscience can tell us about the platform, really, for morality. Now, I want to start with a comment by Ed Wilson in 1975. And what he said was that the evolution of human sociality is the fundamental conundrum of biology. And I think what he had in mind there was the fact not only that we tend to live in groups and so forth, but that there is this phenomenon whereby an individual can behave altruistically. That is to say, and this will be a very simple definition of altruism, but where we incur a cost to ourselves in order to benefit another. Now, this seemed puzzling for a number of reasons, and I'm going to approach it first of all, in a very fundamental biological way. And that is that the fundamental value that is embodied in the nervous system of all organisms is self-care. That is, if an animal doesn't see to its own food and warmth and safety, then it's apt to become food for something else and hence would not pass on its genes. And so everything has the wiring to see to its own well-being and survival. Now, this prompted various biologists, for example, Richard Dawkins, to think that altruism then was really a problem, that we couldn't make it consistent with this fundamental wiring for self-care unless we took the view that altruism is not, as it were, natural, but has to be kind of learned and required of children. And he said this must be the case because we are born selfish. Now, as this is a Darwin event, it's very interesting to me that Darwin took a very different view. And in 1871, in The Descent of Man, Darwin asked the question, where does our moral sense or our conscience come from? And he proposed three components. He thought that we have social instincts. Secondly, he thought that we develop habits and skills as we learn to get along in a social environment. And that thirdly, there is a component of problem solving, especially if the ecology changes in one way or another, and we have to deal with such things, for example, as famine. And that norms which may have been customary for a long time may shift and change, or new norms may arise. Now, interestingly, Darwin was not the first to have this view. And in fact, we can see the heart of his idea actually in Aristotle. Now, Aristotle was really interesting here, and this comes out, I think, also in Darwin and in the new data from neuroscience. And that is that having a particular rule is often very counterproductive in in social groups. And that's because social life is incredibly complex, and no rule is ever really up to the unforeseen complexities that may arise. Aristotle understood that very well, and so he took the view that we learn aspects of social skills and habits as we grow up, but what we're learning is not so much specific rules as ways of getting along that depend on our experience. Confucius also took that view, as did David Hume and Adam Smith. Now, what we, of course, did not understand at the time Darwin put it so clearly was what the mechanisms of each of those three components to the story, what might those mechanisms actually be? Now, what is very new is that many sciences have now contributed to our understanding of the phenomenon of sociality and to the mechanisms whereby mammals and birds are very social. And I'm just listing some of the sciences that are involved here. And neuroendocrinology is the science on which I will draw most heavily. Now, the ethologists, those who study non-human primate and other animal behavior in the wild as well as in captivity, ethologists have been extremely important. And one of the things that they have done is carefully document the kinds of social behavior, which, if seen in humans, we would naturally say is moral behavior or social behavior. Consolation after a defeat, reconciliation after a squabble, pro-social choice, meaning something like sharing food, for example, orphan adoption, which is a very heavy commitment, is seen in the wild by, in 5 distinct cases, by male chimpanzees unrelated to the orphans they adopted. We see examples of empathy, of third-party punishment, fairness, self-control, cooperation, and reasoning. For many of us, as perhaps young philosophers, I think we may have been taught that only humans would exhibit this behavior. And it's very clear that although there are differences between our expression of, for example, pro-social choice and that of a chimpanzee or a wolf or a beaver, in no way they show pro-social choice. In this slide too, I just want to really remind us of something that we all understand, and that is that hominins, not just Homo sapiens, but all of these other wonderful guys, have been around for a very long time. On the vertical axis, we see years in millions, and on the horizontal axis, it's not really an axis, it's just an indication, we see spatial extent. Now, Homo sapiens have been around only about 300,000 years. Genetically, we've changed a little bit in that time, mainly with issues having to do with digestion, principally with milk digestion, and also with issues having to do with skin color and hair color. Homo erectus, interestingly, has been around for 1.8 million years, all of them now extinct. They started off with brains about 1/3 the size of ours, and they ended up, probably as a result of interbreeding with some of these other hominins, they ended up with much larger brains. But it's very interesting to see that these individuals living in small groups had controlled use of fire. They had to have been successful enough to last for 1.8 million years. And they had to have a kind of social organization, which may have been rather similar to what we now see in chimpanzees. And probably for Homo sapiens 300,000 years ago, we too had social organizations, not too much different from what we now see in chimpanzees. Now, the question of sociality that Ed Wilson raises is especially interesting because we know that there are many species of social insects and there are social fish. And so the question that we want to raise now is, so what's so different about mammals? And part of the answer is that our behavior is just a whole lot more flexible and not quite so tightly tied to the genes as it is in the case of insects and fish. And there is a reason why that's the case. So to explain both this kind of sociality that we see in mammals and to explain why we have this flexibility, we need to go back about 200 million years ago to a very surprising evolutionary development. And that is the capacity to generate your own warmth, to be an endotherm, to be warm-blooded. This, of course, is a tremendous advantage for a creature because it means it can forage at night when everybody else has to wait for the sun to come up to get the energy to begin to move. So it's a tremendous advantage, but there is a cost. And the cost is that gram for gram, a warm-blooded animal has to eat 10 times as much. It has 10 times the calorie requirement. Now, that's a huge evolutionary constraint. And you know that. If you have a lizard, you can leave it for a week and it's not going to be very troubled. You can't do that with a dog or a cat. So what did Mother Nature do? And the answer is cortex. And cortex is a brain structure that enables the animal to learn about its environment and to make very clever and interesting choices. So here is a sketch of cortex, and on the left, what you're seeing is a slice through an actual human brain, and the cortex is just this bit in purple on the outside. Now, when you take that bit in purple on the outside and you put it under a microscope, what you see is a most amazing structure, highly organized with very specific cell types on very specific layers. It's a 6-layer structure, and we see nothing like it in reptiles or fish or insects, nothing like it. Birds, this is just an aside because it's kind of a complicated story, but birds were long thought not to have cortex because when you look at a bird brain, you don't see anything quite like this lovely 6-layer structure. But it turns out that birds by convergent development have something very similar. It's just that you have to know how to look and it doesn't look quite so neat. The evolutionary story whereby suddenly we have this remarkable structure in mammals is not understood. We don't know how you get from reptiles with essentially maybe a 2-layer cortex if you're generous, but really not. How you get from that to this magnificent thing. Now, cortex is a great thing. And what is so remarkable about it is that the more you have of it, the better, the smarter you are and the more you can learn. Now, the thing about learning is, however, that when you learn something, not just to hold it in your mind for 20 seconds, but when you learn it and can bring it back, say, the next day, there has to be structure built in the brain, and in particular in the cortex. So that means cortex is very expensive because it involves building so much structure. And indeed, in order for you to learn all of what you need to learn after you're born, when you're born, your cortex has to be very immature. And in this slide, what you can see are neurons in cortex. And in the newborn, you can see they're very slim, very spare, and they elaborate the dendrites, the tree-like structure, and the axons. By 1 month already, you can see the emergence of structure. By 2 years, there's a lot of structure. And at 2, there's actually a little bit of pruning back. And in adults, again, we see that there is still room for learning and structure is still being built. Well, this is dandy, you might say, and what a smart thing for Mother Nature to do. But of course, there is a downside. And the downside you can infer as soon as you look at this slide of newborn cortex. The newborn, and we all know this, of course, the newborn is helpless. And here you see neonate rats, just born. They are blind, they are hairless, they are utterly dependent on the mother. And all they can do is scrabble around, find something warm that sticks out and suck on it. And of course, they do develop very quickly. And rats are, although we find them a nuisance at times, you have to admire their capacity for hunting, surviving, and keeping themselves together. All right, so Mother Nature has a problem. She's going to invent cortex so people can be smart, so mammals can be smart, but you can't just do what a mother turtle does, which is lay your eggs, leave, and the turtles come out and take care of themselves. So when learning capacity goes up as a result of cortex, neonatal independence falls. So what to do now? Who is going to take care of these potentially smart babies? Now, I'm putting it in this anthropomorphic way, but you guys all understand, of course, that there is no such thing quite as Mother Nature. But in any case, the solution to the problem of neonatal dependence is very interesting. And it's the key to understanding sociality in general and morality in particular. So essentially what had to happen was, you remember the slide where we talked about the orientation towards self-value, towards caring for yourself. The wiring in the old structure of mammals was changed, so that essentially what happened was, as the baby was born, the mother's care ambit expanded so that the baby was kind of like part of her, part of herself. And just as she takes care of her own warmth and food and safety, she takes care of the warmth and food and safety of the offspring. That at a functional level is essentially what happened when the wiring in mammalian parental brains changed. First of all, in females, but of course it also changed in at least some males. So that's the conceptual event. What actually is the mechanism? Now, there are many, because brains, of course, are very complex in mammals, but in all things, there are many neurochemicals involved. But one in particular I'm going to focus on here because it really is the crux of the story. And that is this rather simple peptide called oxytocin. And oxytocin is important for delivery. It causes contractions of the uterus. It also is important for milk ejection. And those are things that happen outside of the brain. In the brain, what happens is oxytocin binds to very specific structures in the brain, with the outcome that the mother sees this infant as the most wonderful, beautiful thing in the world, becomes absolutely bonded and attached to this baby. And as the baby suckles and is cuddled and cared for, similar events happen in the wiring of the baby. The baby becomes tightly bonded to the mother. And this is the role of oxytocin. And oxytocin has been known to be important in mother-infant bonding for some time. And nobody really saw that there might be a connection to morality. But just so, because other parts of the story are about to come in, here is a sketch of the human brain. Right here, this is my little fake laser pointer. Right here is the structure from which oxytocin is released. And it's shown in green, rather larger here. A tiny little structure in the ancient part of the brain called the hypothalamus. And it sends out oxytocin to a number of regions. And you can see that here. It sends oxytocin to the brain. It also sends oxytocin to the posterior pituitary and then onwards into the body. All right, so oxytocin is an important part of the story here. How do we get to morality? And the answer is really quite interesting, which is that once this hugely deep organization is in place, whereby you become attached to another and you care for another, that structure can then be pushed a bit this way and pushed a bit that way and modified over here and modified over there. And it can turn out that you can have caring not just for your own babies, although that will be very, very strong almost always, but for friends, for kin, and for mates. So here's the story of the voles. And this was what made me really understand that the circuitry for mother-infant bonding is the circuitry that is also underlies sociality in general. In other words, now I began to see that we can answer Ed Wilson. So a montane vole lives amongst rocks and in the mountains, and it builds its nest under the rocks, and the male and the female meet, they mate, and then they carry on independently. He's looking for more action. She's going to have the babies. It turns out that prairie voles are very different. So male and female virgin prairie voles meet, they mate, and now they're bonded for life. The male helps take care of the babies. He guards the nest against intruders. They like to hang out together. And if they are separated, they show signs of depression. And in voles, what that means is if you drop one in a pail of water, it just kind of, oh God, I guess I can swim, but I don't really have my heart in it. And when you put them back together, they're joyous, they lick each other, and they cuddle together. So the behavior is quite distinctive. And the question for neuroscientists was straightforward. What's the difference in the brain? And it turned out that there is a very clear answer. What you're looking at here are slices of brain from, pardon me? I wonder if we should get rid of this band of photos at the top because part of my slide is missing. So can we just get rid of that? Anyway, it doesn't really matter.
I'm not sure if I can.
It's okay. I know what's there and I'll just talk it through. So what's on the left-hand side? What's on the left-hand side are slices of montane vole, and what's on the right-hand side are slices of prairie vole. And in the top panel, you're going to stain for oxytocin receptors. That is the little protein structures on a neuron to which oxytocin binds and changes the behavior of the neuron. And down here, you're going to stain for something that's very close to oxytocin and also plays a role, namely vasopressin. Now, the stain shows up as dark gray. And what you can see is that in this region here, the prairie vole has stains for a lot of oxytocin receptors, and the montane vole does not. And the prairie vole has a lot of staining down here for vasopressin receptors, and the montane vole does not. And what are these areas? They're part of the reward system. And so what we think, just on the basis of this much science, is that the reward system plays a really important role when oxytocin receptors bind there. In other words, maybe it makes the prairie vole feel good. And that's indeed part of the answer. So the parts of the reward system are called the nucleus accumbens and the ventral pallidum. And OTR means oxytocin receptors, and they are linked to the rewarding aspects of bonding. That's why the male and female prairie voles, after mating, stay together. It feels good to be together. Now, the story is a little more complicated than that, but that's the fundamental part of the story. Here I want to show you, this is a sketch of a rat brain, and this is that part of the hypothalamus that contains oxytocin, and it sends it to the VTA, which is part of the reward system, and there's the nucleus accumbens, part of the reward system. And the nucleus accumbens, you'll be happy to know, is full of receptors, full of neurons that release endocannabinoids and endogenous opioids. And that's why you feel good. And in the next slide, what you'll see is with these little funny things are receptors for oxytocin all over the brain. And what you can see then is it's not just in the old part of the brain, that is the hypothalamus and the reward system, where you see oxytocin receptors, but distributed very widely. Now, probably this means then that there are many areas involved when an animal is engaged in social behavior of one kind or another. So I suggested that the basic underlying mechanisms for mother-infant bonding can be shifted and changed a little bit in one direction or another. And we see in the case of prairie voles that it was changed in such a way as to result in long-term pair bonding. But only about 8% of mammals are long-term pair bonders. Wolves are, beavers are, and humans, although we live very long and it may be serial long-term bonding, humans tend to be. Having said that, of course, we know that in all species there is variability. So we're talking about a predominant behavior, not one that is absolutely universal. But we know that in other kinds of social animals, indeed other kinds of non-human primates, there can be very, very strong bonding between kin. For example, in baboons, the mother-daughter relationship is very strong. And there are, in the Hamadryas baboon species, there are what they call matrilines. So there's a high-ranking matriline where it's the mom and her daughter and her daughter and her daughter. And then there's another matriline and so forth. So there can be different ways in which social behavior gets organized as a function of the ecology and the history of the species and so forth. You'll be interested to know that in birds, about 98% of the species are long-term pair bonders. And ecologically, I mean, there's a very Darwinian explanation of why that would be so. Because if you're a mother bird and you're sitting on your eggs and you're getting hungrier and hungrier as the day goes on, you got to leave the nest and get food. And if you don't have somebody there to guard the nest, your eggs are in trouble, or later your fledglings are in trouble. And so it makes perfectly good sense that so many bird species are long-term pair bonders. So I want to just say a few things about oxytocin. We know that it plays this very important role in bonding. We know that it also affects other parts of the brain and its systems. For example, one of the things that happens, and I think this is terribly important in social behavior actually, is that when oxytocin levels go up, cortisol levels go down. So that even apart from any effect on the reward system as such, when cortisol levels go down, then your stress goes down, your anxiety goes down, you feel better, you feel more pleasant, then it's easier for you to do things like cooperate, share food. And then once that happens, then it's easier the next time to cooperate and share food. And these chimpanzees here, of course, are the ladies sharing food. The determination of whether or not oxytocin is released in food sharing really caught somebody's imagination. And so the idea was maybe in wild chimpanzees, if you collect their urine after they've engaged in food sharing, will you notice an increase in oxytocin levels? And the answer was yes, but I think you should be careful about that finding, because levels of oxytocin in the brain and levels of oxytocin in the body are largely uncoordinated. So I would be careful about inferring too much about this, except for the fact that food sharing is something that very clearly animals, when they do engage in it, does seem to have a bonding effect. And by the way, we see that in rats. All right. So I want now to just sort of step back and then ask the question, as Darwin did, where does our moral sense or our conscience come from? And the answer seems to be that sociality, starting of course with the parent-infant bonding, begets other caring. And other caring together with social learning gives rise to customs and conventions and ways of doing things that are considered by those individuals to be right and wrong. And the social learning, I think, is very, very complex. We learn by example, we learn in humans, we learn in stories and in songs. We learn sometimes by being told a rule, but we all know very early on that even though we are told never ever lie, that when grandma says, "Does my hat look terrible?" you have to lie, right? You have to say, "Actually, it's kind of cute," or something. But every child knows this. And that's one of the reasons why it's been very difficult for philosophers who've been wedded to the idea that got to be a single rule or a single rule or two, the Kantians and the utilitarians, why they've had such a bad time. In this slide, this is just really to remind you of the importance of cooperation in a very social species, namely these are gray wolves. And watching the predation involved in gray wolves is really interesting. You know, they're not successful quite as often as stories might indicate. And so they're often quite hungry. And what's really critical, I think, in watching how they operate is what we see actually right here is the distribution. They all know what's expected of them at the right time, and they'll read this in the tiniest little bit of behavior. This guy is going to go and rip the back leg out, and this guy are going to come in and take the throat. And they're very efficient, and they modify their behavior, their predation behavior, as a function of circumstance. It's not just doing the same thing every time. They read each other, they read the prey, and their social brains are really, really impressive. Incidentally, the male and the female are the only breeding pair in the pack, and they do bond for life. And the dad wolf takes intense care of the young along with the female. And here too, in very exceptional and very difficult circumstances, we see that cooperation is essential for survival. And here are, in 1903, a group of Inuit in the Arctic cooperatively building an igloo. The children are not explicitly told that they have to cooperate and that they have to get to work and they have to do one thing or another. They imitate, they do what others do, and it feels good, and they get rewarded for doing the right thing. And so, when I think about that funny comment from Richard Dawkins about how you really basically have to beat decency into kids, I'm thinking, no, basically not. Now, there are difficulties with certain kinds of individuals, the ones that when they're adults, we call psychopaths, but that's relatively rare, and we can talk about that later. So we've talked about the importance of the hypothalamus and the release of oxytocin from the hypothalamus to wide regions of the brain. It's also recently been discovered that in addition to what's sometimes known as the where pathway in the visual system and the what pathway in the visual system, there is a large pathway shown here in red that is specializing in detecting and understanding the meaning of social behavior. It's this pathway that allows those wolves to read a tiny signal, an ear twitch, a head movement, a tail movement, a tiny signal, or in some cases, a large signal, and to know what it means and to integrate it into your behavior. And in this slide, we can see the importance of, these are all rather exaggerated facial expressions, but we instantly have no trouble reading. I mean, you don't have to say, oh, well, gee, why is he showing his teeth like that? You know that this baboon is very serious and does not want you around. And similarly here and here, these guys, it's within milliseconds. You know that they are all highly focused visually on something in their environment that matters to them. And we know by this behavior that these guys are alarmed by something. So that's the part of the visual system that's specialized for this. So when Ed Wilson asks about social behavior, what we're now learning is that there are large parts of the brain that can do other things, can change a tire perhaps, or pick pears, but that large parts of the brain are sensitive to social signals. And in this slide, this is very recent work that came out of Michael Platt's lab, showing that in frontal structures, prefrontal structures, the orbital frontal, which is just above the orbits of the eyes, the dorsolateral, have neurons that are highly sensitive in natural conditions to the value of what's going on. So we evaluate the meaning of a social signal. Now, since I have a little bit of time left, I really want to talk to you about this slide because one of the things, of course, we're very interested in is social behavior where something seems to have gone wrong. So in this lab, what they were looking at was the importance of early experience in baby mice. So mano just means zero manipulation. So the baby mice are taken from their mothers with even the experimenter doesn't touch them, picks them up in a cup and puts them in a place where they're warm and fed and safe. So they should be fine. And at the end of the day, they're actually brought back. So they spend quite a lot of time as tiny mice, babies away from the mother. What do you see? And here's the critical part of the story. What we see is that part of your DNA that codes for the receptor for oxytocin, that part is shut down. It's methylated. And that's important because it means that if your DNA as a baby mouse is not making a lot of oxytocin receptors, you are going to have a system that's very different. So we see decreased gene expression for oxytocin receptors because it's been shut down. And consequently, we see decreased binding to oxytocin receptors because there's fewer oxytocin receptors to bind to. And then the question is, so what happens to the social behavior? And you see decreased species typical social behavior. And in particular, what you see is that the female mice who were in this experiment, when they have babies, are very poor mothers. They really don't care. They don't bond to their babies. They don't stay with them. They don't protect them. They're very poor mothers. So this is a really important story. We don't know whether this happens in humans. It's certainly a leading hypothesis that it may, because this mechanism, this system for mother-infant bonding is so old, so deep, that it's likely to be very similar across mammals in general. So one question I raised earlier had to do with psychopaths, those who appear not to have any feelings of social care or social concern, don't form really lasting social bonds of any kind, can be very manipulative. They may understand social situations quite well, and that may allow them to exploit the kindness and goodness of other people for their own benefit. Work on this, of course, is important. And we know that there is a strong genetic connection. That is, that if one of your parents is a psychopath, that increases the probability that you may be also. So we see polymorphisms in psychopaths in the gene for making receptors for oxytocin. But this is still all early data, and doing these kinds of studies in humans is very complex for all kinds of reasons. One thought that occurred to people once the story on oxytocin and sociality in voles came out was, well, maybe we can make people more social, or animals more social, by getting oxytocin into the brain. How might you do it? Well, think about cocaine. You just sniff it, goes up the nose, goes into the brain, it has an effect. Maybe we can put oxytocin into an intranasal spray and shoot it up the nose and see what happens. There were some early reports of really quite interesting effects, but it was all kind of head scratching, because it turns out that there is this thing called the blood-brain barrier, and only some things cross the blood-brain barrier. Does oxytocin cross the blood-brain barrier? It looks like not. And the work showing this was really quite clear. And but then somebody did the work on monkeys, and it turned out that just in two instances, they sprayed oxytocin up the nose, and by gosh, they found it in the monkey brain. So the question was, how the heck is it getting in? And does it get to a place that really affects social behavior? Now, it turns out that on the roof of your nose, there is a thing called the cribriform plate. And there's little teeny weeny holes in it. And the theory at the moment is that oxytocin goes up through those little holes and into the olfactory bulb. and it kind of drifts along the outside of the olfactory nerve and maybe drifts around into places in the brain where it will have an effect. But the great hope that we might be able to counteract certain kinds of social disorders by using intranasal oxytocin now looks much more difficult than it was initially. So again, just to sum up the idea behind this whole story of oxytocin that starts with being warm-blooded and involves infants who have to be cared for, is that sociality, the mechanisms for sociality involve caring for others, whether those others are infants or whether they're friends or mates or kin. And that other caring together with learning and acquiring the skills to get on in your social group give rise to social norms that we feel very deeply. We feel that certain things are just wrong. And finally, that problem solving in an ecology begets norm changes. I think with regard to neural mechanism, the third is the one that we understand the least. But of course, as Aristotle and Confucius and David Hume too made very clear, a single rule for all cases never works. And it doesn't, because norms conflict with what we want sometimes, with our preferences. And that conflict can vary as a function of whether you're well-fed or starving, or whether you're exhausted or you're energetic. Norms conflict with other norms. Norms vary across individuals and across groups, not hugely, but to some degree. And norms vary within an individual across time. And one of the things that is really puzzling is how the brain pulls together the relevant memories when we are in a moral dilemma trying to figure out what to do. How does it know which memories are relevant to helping us solve our problem? Finally, and I'm sure this is something Darwin would have understood, is that given the nature of the pleasure of socialities, many species, ours in particular, of course, derive great pleasure from interacting with other species. But we are not unique in this. And so we know of examples in the wild where the circumstances permit animals from very different species, in one case, for example, a black bear and a cat, to get along, to be chummy, to cuddle together, and to like each other. So we may see many behaviors that are largely unrelated to passing on our genes. But I think we're doing well enough passing on our genes that we needn't worry too much about that. So I've drawn mainly on the work that I discussed in this book. This is the most recent book. And I would be more than delighted to get your questions and thoughts and concerns. And thanks so much for inviting me.